Monday's video looked at the first level of gene regulation in eukaryotes, and we looked at modifying the chromatin structure. So is the chromatin tightly packed or is it loose? Another way to think of that is, can polymerase get to the gene or can polymerase not get to the gene through transcription? So think of that as phase one. Now, level two, we're going to be looking at regulation of transcription initiation. So flashback to when we looked at transcription initiation. That involved the attachment of transcription factors to the promoter, to the TATA box, and they acted like signals to help RNA polymerase get to the gene. If you remember my ridiculous example, that was like those crazy things at car dealerships that call your attention, right? So you can picture me doing that. That was kind of a lie because it's a lot more complicated than that. So that's what we're going to look at today is how is this actually happening and how does the cell regulate that? So before we jump into the molecular stuff, we want to talk about what's turning this gene on or what's turning this gene off. We really just want to think of this as on being more accessible to polymerase and off being less accessible to polymerase. So that's what we want to think about when we talk about on versus off in this example. Now let's look at our typical gene. All of this you should be familiar with. I am going to block out all of this nonsense because it's overwhelming when you look at all of it. And we're going to focus on the gene. So you know the promoter and you know the gene, right? That's all what you know. The polymerase will jump onto the promoter and then move down the gene doing transcription, and then we end up with our primary or our pre-mRNA. That's nothing new. What is new is what's going on to the left of the gene. These yellow dots or spots on the DNA are what we call control elements. They're just specific sequences of DNA. You can almost think of them as little mini operators like we saw in the operon system. They're just little segments of DNA that are going to play a role in the control of this particular gene. And like the operon, we're going to see that something attaches to these control elements that's going to make the polymerase attach to the promoter and move down the gene, turning that particular gene on. So take a minute and just kind of review this figure. So what's a promoter? Where's the gene? And then what actually is a control element? Now again, let's just review what is a transcription factor. Transcription factors that we looked at before were those proteins that attached to the promoter that called or signaled to RNA polymerase. For technicality, we're going to call the ones that you already know general transcription factors. Those are the ones that attach to the TATA box, call the attention of RNA polymerase, and allow transcription to start. But there's another type of transcription factor that we call specific transcription factors. Those are going to be the substances, the proteins that attach to these control elements. So what you're going to see coming up is that transcription factors, the general ones, are going to attach to the promoter just like we've seen before. And then other specific transcription factors are going to attach at these control elements. All of that attaching is going to be a really big signal to RNA polymerase to say, hey, this is where you need to do transcription. Okay, so let's take it one more step further. So regular transcription factors happen here, and then our specific transcription factors attach there. Now, this picture is very scary So we're going to take it a little bit at a time, and then we're going to look at the animation to see this a little bit more in motion. So what this picture is showing you is this original sequence that was on the previous slide. We still have the gene, we still have the promoter, and then we have our control elements upstream. What you see going on here is these specific transcription factors, the special ones called activators, are attaching to the control elements. The transcription factors are attaching to the promoter. So you see that it's going on down here. Ignore the mediator proteins. We're not necessarily going to get into that. It looks like a thing of grapes. We're just worrying about the specific transcription factors called activators and the general transcription factors that you already know. When these things all attach to the DNA, so the activators are on there, the general transcription factors are on there, that allows the polymerase to bind to the DNA, and weird thing that happens, the DNA bends over on itself, and when it's all attached like this activates the polymerase. So let me show you a quick little clip of that. Unfortunately, I can't give you the sound through here, so I'm going to just talk you through it, but it shows this a little bit more in motion. Okay, so what you're looking at is a different diagram of the same concept we were just looking at. So we have our enhancers, we have the promoter, and then we have the gene that needs to be transcribed. The goal of this is we want RNA polymerase to be able to attach and transcribe the gene. This is where our transcription factors come into play. These transcription factors are those activators from the previous slide, and the pink ones are the regular general transcription factors that we all know and love. So what he's saying is that the general transcription factor is attached to the promoter. That's going to signal RNA polymerase to come in, but it's almost like it's frozen. It can't get started until all of the transcription factors are attached. That's where that specific transcription factors, those activators, come in. That causes the DNA to bend over on itself. And that almost gives the RNA polymerase a little push or a little activation to go through and transcribe that gene. So as you know, that gene would be on, making the mRNA, and then eventually we're going to make the protein. So I will post that Bozeman video on my website so that you can watch it again and maybe hear someone else's perspective. Do you necessarily need to know all the details of the mechanism of this? No. At the very bare minimum, I would say know that there are different types of transcription factors, those that attach to the promoter, those that attach to the control elements upstream of the DNA, upstream of the gene. And all of those binding of transcription factors helps the RNA polymerase bind to the gene and initiate transcription. Now, why am I introducing these activators? Let's take it back to kind of our big point that we hit on yesterday. In a multicellular organism, we have cells that are differentiated. We have cells that are specialized and have their own jobs. That comes from the fact that even though different types of cells have the same DNA, in that cell, some genes are turned off and some genes are turned on. And those activator proteins are going to be one of the ways that this differentiation can occur. So we're going to look at a liver cell and then a lens cell, so the lens of your eye. And we know that regardless of what type of cell it is, these two cells are going to have these exact sequences of DNA. This sequence of DNA contains an albumin gene. Albumin is a protein that the liver uses to function. And this segment of DNA has a gene for crystalline. Crystalline is a protein that helps your lens function. So just thinking through this logically, in the liver, would we want an eye lens protein? No, it's not going to have any function in the liver. In an eyeball cell, so in the lens, are you going to need a protein for liver functioning? No. So this is where differentiated gene control comes in. In the liver cell, we're going to see certain genes on and certain genes off. And the lens cell, we're going to see different genes on and different genes off. And that all comes from those activator specific transcription factors. So looking at the left side first, the left side has these particular activators present in the cell. Those ones fit perfectly with the control elements that are with the albumin gene. So they're going to attach, all the transcription factors are going to attach, and then this gene is expressed. Now looking at the crystalline gene, we would need orange, gray, and pink. Those things are not present in a liver. So there's nothing to attach to the control elements. So this gene would never be expressed because polymerase would never find it. Okay, now looking at the right side of this, in a lens cell and an eye cell, again, on the albumin gene, we would need a yellow, a gray, and a red. But why would a lens cell need to express a liver gene? It makes no sense. So those activators are not there. We have gray, orange, and pink. But our crystalline gene has control elements that would attach with orange, gray, and pink. Since those activators are present, they're going to attach to the control elements, get polymerase all situated on the promoter, and then this gene would be expressed. So the big idea that I want you to see and I want you to focus on is that this whole crazy mechanism of activators and control elements is one of the ways that our cells can control which genes are on and which genes are off. And we just used the example of liver cells versus lens cells, right? This cell is going to want certain genes to be expressed so that the liver can do its function. But in contrast, an eye cell would want different genes to be turned on in order for that cell to function. Again, do you need to know the nitty-gritty details of all of this fanciness? No, but I kind of gave you the background information so that you could understand what was going on in this figure. Okay. Try to review that a few times. Watch the Bozeman video. I will post that as well with the sound. If you have questions, let me know.